Hi everyone, thank you for coming. Today I'm going to tell you about a project looking at a translational polygenic score of biological sensitivity to context, which is a theory depicted here in the schematic and suggests that there's a subset of individuals that have a high malleability or plasticity to the environment, and they have a lower likelihood of developing a psychopathology in positive contexts, but a higher likelihood of developing a pathology in negative contexts compared to individuals with low biological sensitivity to context. We wanted to test this theory empirically, and we did that by using a translational model beginning in rodents, using RNA sequencing from the ventral dentate gyrus and identifying genes that are significantly upregulated or downregulated in animals exposed to environmental enrichment compared to controls, or animals exposed to social defeat compared to controls. We then had two distinct groups of genes, upregulated and downregulated in these different contexts, and we compared the list to identify genes that were commonly upregulated or downregulated, suggesting that they're responding to the change in the environment rather than the valence of the environment itself. We then, in the end, had a grouping of environmentally responsive genes, and we wanted to use these genes to test whether or not we can identify individuals with high biological sensitivity to context. And the way we did that is by using a translational model identifying the human orthologs of those genes and building what we call a polygenic score, which uses GWAS summary statistics to obtain a single value estimate of an individual's propensity to a phenotype. In this experiment, we use the GWAS for major depression given the implications of social defeat in MDD. In the end, each individual received an environmentally responsive polygenic score, or ER-EPGS, as you'll see. We then split the sample by the median such that individuals with a high polygenic score should show high biological sensitivity to context, and those with low environmentally responsive polygenic scores should show low biological sensitivity to context. And that is depicted here. This is our hypothesis that individuals with high polygenic scores should be very plastic and respond to both positive and negative environments. But in order to test this theory appropriately, we needed to build an environmental score that encapsulated both positive and negative aspects of the environment. So we built this score for each individual in each cohort. You'll also see red lines on my graphs depicting where in relation to the interaction the significance lies. These are the four cohorts we tested our theory on: MAVEN, GUSTO, ALSPAC, and UK Biobank. MAVEN and GUSTO are both cohorts of children. ALSPAC is in adolescents, and UK Biobank is in adults. You'll see that ALSPAC and UK Biobank are very large studies, so we were able to further separate them into males and females. When we look at MAVEN, we see that individuals with a high polygenic score show high anxiety problems at 5 years old, but only if they're exposed to these adverse contexts. That was really interesting, and we replicated this finding in GUSTO, identifying that individuals with a high polygenic score have high anxiety problems in adverse environments, and also identifying that these individuals, or individuals with a high polygenic score, have low anxiety problems in buffering environments. When we looked in ALSPAC, we identified that the outcome changed to depression when the emergence of these of depression and other psychopathologies arise. Individuals with a high polygenic score had high depression scores in adverse contexts, but low depression scores in buffering contexts. This effect held in females, but did not hold in males in the ALSPAC adolescent cohort. In adults, we see that individuals, again, with this high polygenic score have high mood and neurotic disorders in adverse contexts, but low neurotic disorders in buffering contexts. When we split this further into males and females, we identified that this effect, again, held in females, but not in males. In summary, today I showed you that we built a mouse model to identify common patterns of gene expression in the ventral dentate gyrus. We used these genes to build a polygenic score and identified that this score moderated the relationship between the quality of the environment and psychopathology in 4 distinct cohorts. Thank you for your time.